This comes out of a commercial lab, Spectrum Analytic. The principles that we've been talking about in terms of taking accurate composite samples are covered again in this same presentation. We won't go over them ad nauseum, but making sure that we've got a good composite sample really is dependent upon making sure that we're getting it from a representative area and making a good physical average. And so the same principles apply. If we have anomalous areas, we avoid them. If we have larger anomalous areas, we may want to sample them separately to make sure that we're getting an accurate idea of what's going on within those different areas. But when it comes to plant sampling, the compositing is of the same nature, except it's very unique to each plant, not necessarily each plant, but each type of plant in terms of where we take the samples. And that's really what I want to kind of make sure that we cover today, along with a couple of things about preparing them for shipping to the laboratory and so forth. So the when and where to take samples we'll cover here in a minute in the table and diagrams in this particular PDF. But the amount of material that we want to end up with is somewhere around a softball-sized conglomerate when it's kind of not compressed necessarily, but gathered. And we want to be able to prepare that properly for sending it to a laboratory. So we'll put them as we take them from the different areas, at least 10 areas within a field. And put them in a large, clean paper bag or plastic bucket. In the case of nutrient analysis on tissues, it's really important that we don't use a galvanized metal bucket or container for this kind of thing to avoid zinc contamination and other micronutrient contaminations. It is common that we'll have dust or residual materials from a foliar application or other things, excuse me, on the surface of the leaf. Those can be removed with a clean rag, or even they say a soft, dry bristle brush, but that might be a little aggressive in most cases. But really, in most cases, we don't want to overwash a sample because we don't want to leach plant tissue of any important nutrient content that we have there. Make sure that the samples are dry to the touch before we package them for sampling. They recommend in this particular case that they be strongly wilted before shipping and that they're placed in a paper bag and not a plastic bag. That's an important aspect of all plant tissue sampling as they're shipped to a laboratory for analysis or ported to a laboratory for analysis, because we don't want that material to rot within the bag as it might condense and get some moisture and activate microbial activity and so forth. So we want it to stay fairly dry. So paper bags, paper envelopes, and things like that are the best way of getting that to the laboratory. But the main thing about plant sampling is when and where to take the samples on the plant. And that's really what this particular handout is outstanding for, because it breaks down every possible crop that we might encounter. These are our field crops, our grains, and so forth that we might encounter, hay and things like that, and where they are in terms of the stage. If we're taking for instance, in a small grain situation, if we're pre-boot on the grain, then we're going to take samples from the entire above ground portion. We do not include roots in any way in plant tissue sampling. That'll really cloud the results. And 30 to 40 total plants so that we get enough plant material. Beyond boot stage, then we're looking at taking flag leaves. And in that case, we want to take 30 to 40 leaves in order to make sure we've got enough sample in that situation. Corn, the same kind of thing. In the vegetative stages up to V12, we're looking at taking above ground material. Beyond V12, once we get to tasseling, we're looking at a flag year sampling, or just below the whorl where the tassel's emerging. And it tells you how many leaves or plants to sample from in this particular table. Hay, we're looking at the above 6 inches, the upper 6 inches of grass hay. In terms of alfalfa, the same situation, but prior to 10% bud on alfalfa. Vegetables are included in here as well for those of you that have interest in horticulture production for those. Most of those are typically the most mature, fully expanded leaves, and how many leaves that we would be interested in those. We do have occasion to have folks involved in potato, at least in the northern Cache Valley area. And so there's an indication there in terms of the seedling stages, and then once it goes into bloom, how many and where on the plant. So I just wanted to kind of introduce you to this table and this particular PDF publication, because I think it's really a valuable document to have handy when we're looking at the sampling of different plant types. Fruit trees mid-season on fully mature leaves on either terminal or non-fruiting spurs and branches, in canes on primocanes, fully expanded leaves, and how many leaves, etc. So I'm not going to spend a lot of time parsing through this table. I just wanted to make sure that you are aware of it. Not only does it describe them in a narrative way, but it also gives really valuable pictorial guides to where these leaf samples and tissue samples are taken for plants of various stages. So unless there's any questions on what it means by these different stages, they're well illustrated in these graphics. I think all of you ought to print this off and have it handy, and we'll look at kind of modifying some of our available documentation here at Utah State to help reflect some of this, because we are getting more and more interest in a variety of different plants to sample for tissue analysis. And this, I think, is one of the most complete guides that I've really ever come across. And so I'd recommend it to you. So the last few things that you wanted to note: plant tissue samples are really either to confirm a deficiency that we think we have from maybe soil analysis or visual symptoms, maybe see in some cases where we might have some hidden stresses where we can do better, and maybe some of the micronutrients and their management. And it's really geared towards improving fertilizer management, nutrient management in future subsequent crops. There's not much that we can often do with tissue sampling to correct in-season deficiencies, except in some things like cane fruits and others, where we might be able to get some foliar application of zinc or iron in those particular cases that will help correct an in-season problem. But there's a lot of interesting do's and don'ts. I just wanted to note a couple of things to avoid. Don't overwash, don't sample through stress periods, don't sample severely injured plants or dead areas, and do not include roots or woody materials in these, any loose soil and so forth in the samples that might frustrate the analysis in the end.